Hi, I am Cher Wilson. I'm the manager of the microbiology lab here at UNC Hospitals in Chapel Hill. Have you ever wondered what happens to that throat swab that they take at the doctor's office, or that COVID swab? Well, let's go find out. Come and follow me, and I'm going to show you the microbiology lab and what we do here. This is one of the entry points for almost 1,000 specimens a day received here in the microbiology lab at UNC Hospitals. The sample is scanned into our computer system and a unique number is given to each patient and their sample. A label is then printed to be used in the next step. The next step is for the technologist to pick an agar plate that complements the type of test that's being run. Agar contains nutrients for different organisms that help them grow so that we can identify them. The specimen is then added to the agar plate. Once added, a sterile loop is used to perform streaking for isolation, which allows the next technologist, once the organisms grow, to find a single colony that helps us identify them easier. This is the Keystra, also known as Total Laboratory Automation, or affectionately known as the robot. It allows us to process all of those samples faster than we would be able to if we did it by hand like you saw earlier. The robot can take the samples, scan the barcodes that were placed on them earlier. It knows what agar plates to pull. It pulls them. It then utilizes a metal bead and a magnet to create those cool streak patterns and then transports it via an automatic line to incubators where the organisms can grow if they're there, and at intervals pictures are taken, which allows our technologists to look at them further. Once the bacteria has a chance to grow in the incubators, it will take pictures of the plates at different intervals. The technologist can then retrieve the pictures on a computer screen and analyze them to see if anything looks interesting or amiss, something that we might need to investigate further. The other thing that we do is help the physicians know which antibiotic to give. We do that by growing the bacteria on a plate and then putting a strip with antibiotic in it to see if it will kill the bacteria around the strip. This one shows that this bacteria can be treated by that antibiotic. Here is another version of that antibiotic test. This is a green Pseudomonas, and it has multiple antibiotic discs on it. So this antibiotic would be the best drug to give this patient because it killed the most bacteria. Bacteria can have different colors, different size colonies, different shapes of colonies, and those different agars that have different nutrients grow different bacteria. These are some examples of really cool colors. Some more colors, some different bacteria with different colors and shapes. This one has a bullseye in its colony. And this shows you why we did that streaking pattern. So we can streak for isolation and go in and get just one single colony to help us identify which infection this patient has. In the microbiology lab, we don't just look at bacteria as causes of infection, we look for other causes. One of those is yeast. And just like the bacteria, it can have different colors and shapes of its colony. Another one that we look for is mold or fungi. And those can really get funky, like this blue-green one, this white and black spore-laden one, and we keep these lids on to protect us from the spores. This one is yellow and brown and very bumpy. And the last one is just one single black colony, fuzzy black colony, none of which are good. Another type of infection that we look for in the microbiology lab is a parasitic infection, and this is an example. This is an ascaris worm. It is a roundworm, and it is from a patient who was a farmer and did not wear shoes while walking around a pig farm. Another parasite that we see commonly here in the special microbiology lab is a blood parasite that you may have heard of, and it's the parasite that causes malaria. And as you can see up on this screen from this slide that I'm looking at, we have white blood cells. Those are your immune cells. We have red blood cells that look nice and normal, but then we have a blood cell that has something in it. And that is the organism that causes malaria. Well, that's it. I hope you enjoyed your tour of the microbiology lab here at UNC Health, main hospitals in Chapel Hill. We do a lot of really cool work here, as you can see, but we also have a lot of fun. We decorate for the seasons, as I'm sure you've seen during your tour. We celebrate milestones, birthdays, babies, weddings. We have a lot of fun together. So, thanks for visiting.